Hello, everyone. My name is Mingkai. I'm honored to introduce a novel genotyping system for porcine epidemic diarrhea virus, PEDV, based on the polymorphism sites on spike protein gene. PEDV is a major pathogen threatening the global pig industry, mainly causing diarrhea in newborn piglets with high mortality rates. PEDV strains are evolving rapidly worldwide, but the underlying molecular mechanisms remain unclear. Based on genetic analysis, we proposed a new PEDV genotyping system. We also found that different regions of the PEDV spike gene are affected by adaptive evolution and homologous recombination respectively. These two factors jointly drive virus evolution. The highlights of this study are as follows. Through comprehensive data analysis, we applied the polymorphism patterns of amino acid sites to genotyping system and reconstructed the global evolutionary dynamics of PEDV. For the first time, we revealed the unique recombination pattern on PEDV, proving that genetic recombination may play a key role in virus evolution. Our research provides a better understanding of the evolution pathway of PEDV and other related coronaviruses. First, we investigated the molecular characteristics of the prevalent PEDV strains in China. We found that the diversity among strain population is high. Multiple strains were co-circulating and significant differences in genetic homology were observed. This result indicates that more precise classification for PEDV is needed. N-linked glycosylation plays an important role in the biological functions of viral structure protein. We observed 3 N-glycosylation sites with polymorphic characteristics among different PEDV strains. They are in the 57, 723, and 1193 amino acid site of the S protein. It suggests that we can use this polymorphism to classify strains. In addition, we found the cysteine mutation at position 1158 has typical temporal and geographical distribution characteristics. This mutation is commonly present in strains circulating in the Americas, Japan, and South Korea after 2013. Next, we collected genetic data from the NCBI database. We used polymorphic sites to classify 2022 global PEDV strains. Based on the polymorphism patterns of N57 and N1193, PEDV strains were classified into 5 genotypes, namely G1A, G1B, G2A, G2B, and G2c. Further incorporating N723 and S1158 into the framework, a total of 12 lineages, from L1 to L12, were obtained. Adaptive evolution can alert viral pathogenicity and antigenicity. We evaluate the selection pressure on the S protein based on algorithm software. We categorized the strain sequences into different datasets based on time, region, and genotype, and defined the sites that were generally under positive selection in multiple datasets, as strong highly intensive selection sites. We found that the His sites are concentrated in the middle region of the S protein, suggesting that these regions may have undergone more frequent adaptive evolution. In addition to adaptive evolution, homologous recombination is also an important evolutionary pathway of coronaviruses. In PEDV, we used RDP software to detect multiple recombination events. More than half of them involved in the fragment exchange at the 2 ends of the S gene. We further conducted a discrete phylogenetic analysis. The S gene was divided into different recombination fragments according to the positions of recombination breakpoints. We found that G2c strains have high homology with G2b and G1b strains in the D0 region and CD region at 2 ends of the S gene respectively, suggesting that G2c may originate from the recombination of G2b and G1b. Based on background information of different lineages, we reconstructed the global dynamic evolutionary history of PEDV. PEDV first emerged in Europe. During its decades of prevalence in Asia, significant strain divergence occurred. Highly pathogenic strains broke out on a large scale in Asia around 2010 and caused outbreaks in North America in 2013. Among them, 2 potential recombinant lineages, G2cl4 and G2cl10, dominate the prevalent populations in China and the United States respectively. In conclusion, in this study, we proposed a novel nomenclature system for PEDV strains. This system can help us understand the evolutionary mechanisms of the virus and conduct epidemiological research more efficiently. However, unfortunately, although strains of the same lineage may be more similar in phenotype, our genotyping system cannot yet be used to directly evaluate the virulence and immunological cross-protection of PEDV strains. Thank you for listening.